Hello everyone and welcome to today's DFHCC Connect Science Seminar. I'm delighted to introduce today's speaker, Dr. David Livingston. David is Professor of Genetics at Harvard Medical School and the Emil Frey Professor of Medicine at Harvard Medical School. He's the chair of the Executive Committee for Research at the Dana Farber Cancer Institute and the Charles A. Dana Chair in Human Cancer Genetics at the DFCI. David has a storied career, having made numerous important discoveries related to the functions of tumor suppressor genes. He's a leading light in the elucidation of the interaction between tumor suppressor genes and cell cycle control. With an initial focus on DNA tumor viruses, his lab discovered the interaction between the SV40 large T antigen and the retinoblastoma protein in the late 1980s. This led to his lab's identification and elucidation of the E2F family of transcription factors. His lab also discovered the P300 family of transcriptional coactivators. In the mid-1990s, David began working on the newly cloned hereditary breast and ovarian cancer predisposition gene, BRCA1, and made the first connection between BRCA genes and homologous recombination. His lab has continued to play a leading role in understanding how the BRCA pathway and genomic instability mechanisms connect with the development of breast and ovarian cancer. David was elected to the American Academy of Arts and Sciences in 2001. His discoveries have been recognized by numerous additional awards, including most recently the Basser Global Prize in BRCA Research and the Pez Collar Foundation AACR International Award. Before I invite David to begin his seminar, I'd like to remind you to consult the Connect Science website for forthcoming seminars and for recordings of previous seminars. However, I'd like to let you know that this is the last Connect Science seminar for the spring, and this series will resume in the fall. Finally, please write your questions which David will address at the end of his presentation in the Q&A box associated with this Zoom call, the Q&A box, not the chat. And so without any further ado, I'd like to welcome David, whose talk today is entitled Unexpected RNA Dependence of BRCA1 Function.
What do I do? Oh, I see. Well, I'm almost as red in the face as the picture you've all seen that announces the existence of the Connect Science series, because Ralph, who is a brilliant investigator, made, I would say, stellar discoveries in the beginning of this field when a number of us had the privilege of working with him here at Dana Farber. He remains a major, more than major, a leading figure in DNA damage control, and I'm deeply honored that he has introduced me to you today. So to stay on time, I'm going to spend a few minutes reviewing what we kind of as a group need to know to feel our way through a body of new data, almost all of it new data, that is in part published and in part hopefully heading in that direction, which I will start with now. So I think everybody knows that BRCA1 is a major human tumor suppressor. Its germline loss translates into cancer. The classic cancers in certain families over the years have been breast cancer and ovarian cancer, as well as in men, prostate cancer, and in both genders, esophageal and pancreas cancer. And in about 3% of all the breast cancer in the United States, one can find a molecular, even a genetic link to inherited loss of BRCA1 function mutations. And in the tumors that develop in these individuals, the gene, the BRCA1 gene, which may have started its life off as one good and one dead copy, is now, in many of them, not all, 2 dead copies, meaning that a tumor in these classic forms of the disease effectively have no intact BRCA1 operations to lean on. Now, the mechanism of tumorigenesis, which is still a great mystery and one that a number of us are working on, is incompletely understood. But there is a breakdown in genome integrity control that we believe, and I will show you some evidence for it, triggers aberrant epithelial, mammary epithelial cell differentiation. And we believe, and you'll see some evidence that that plays a leading role in the ugliness, the medical ugliness that follows it. The BRCA1 gene is large. It encodes 3 overlapping proteins. The killer protein that we want to talk about is a killer only scientifically. It's the tumor suppressor. It's P220. It occupies, its sequence uses effectively, or is dependent upon the entire coding unit of BRCA1, which is present in a series of exons. And we're not going to spend any time on the other 2 BRCA1 proteins until perhaps the question and answer period, where I'd be glad to give you a little bit of information if time holds up. And the reason for that is there's enough to talk about in the next 50 minutes to keep at least me awful busy. All right, first, BRCA1 is multifunctional. The key fact that I kind of like to wake up in the morning with, because it's humbling, is that BRCA1 has been shown by other labs, Alvaro Montero, Roger Greenberg, and yet others, to bind 250 separate polypeptide proteins, 200 separate polypeptide chains. 250 proteins are the repertoire we know of at the moment that calls itself BRCA1 associated proteins. Right now, we know of 4 generic forms of behavior that BRCA1 itself performs. And in the process of performing it, in some cases where it's been studied carefully, there is a dependence upon BRCA1's ability to bind to 1 or more of these 250 proteins. The 4 generic forms of function that I want to talk about are tissue homeostasis and cell differentiation, which is a BRCA1 function. DNA damage repair is by far the most famous of the various generic functions that it executes. And the ability of BRCA1 to carry out certain forms of DNA damage repair is life-saving in many cells. For example, double-strand breaks that arise spontaneously or otherwise need to be repaired perfectly, and BRCA1 is kind of the leading advocate, if not the leading player, in that process. You also need to carry out proper cell division in order for a normal, for example, mammalian cell to carry on. BRCA1 plays a role in proper cell division. And lastly, it has a burgeoning role in regulating transcription and RNA biogenesis. And as you'll see, I'm going to focus the first half at least, maybe more, of this talk to a new set of functions that have unveiled themselves in our group by a small cadre of really talented investigators led by Elodie Hatchie, a senior member of the unit here at Dana Farber, about whose work we will talk. All right, here's a little follow-up and a summary. So let's talk about DNA damage repair. I scrawled on this little sheet of electronic paper a diagram of the sequences and domain structure of BRCA1 P220. That's the tumor suppressor. And you can see there are a bunch of bound proteins that are referred to. A protein called BARD, which is actually a cousin of BRCA1 itself. The MRN complex, famous for participating in a very important early set of steps in homologous recombination of double-strand breaks and so forth. You will notice a few domains, a ring domain at the western end of the protein, 2 nuclear localization sequences that we're going to talk about in detail, hopefully in about 30 minutes, and hopefully not much longer. And then a coil-coil domain, a bunch of phosphorylation sites, and then a pair of imitators. They're called BRCT motifs. They're famous in DNA damage control as being components of important DNA damage control proteins. We'll talk about the BRCTs as well. And then there are a number of associated proteins that are written down here, which I don't want to waste your time going through, but all of them at some level play a role in DNA damage resolution. So let's talk about DNA damage functions in BRCA1 when the protein is at least heterozygous. That means if one looks at the genes as being simply inherited as opposed to somatic, BRCA1 plus minus cells are normal at carrying out homologous recombination type perfect double-strand break repair. They're haploinsufficient for stalled fork repair, which we're going to come back to. That's a relevant finding. They're involved in interstrand cross-link repair, which we'll also talk about. And then there's this issue of BRCA1 and RNA. And in that case, we're going to talk about the ability of BRCA1 to confront I guess you'd call it, it's like an engineering artifact in the genome, which is the formation of R-loop structures, which are RNA DNA hybrid-driven loops with a single-stranded DNA and, of course, a single-stranded RNA loop to comprise a very healthy section of it. These are abnormal structures when they form a number of genes because they promote the development of DNA damage. We're going to say quite a bit about them. And then in the end, let's talk about the most well understood BRCA1 disease, a disease that arises when BRCA1 fails its job as a tumor suppressor, and that's cancer. And you can see it's even possible with one active copy of BRCA1 and one dead one to arrive at a tumor whose genotype for BRCA1 is heterozygous, even though the more famous genomic development in cases like that isn't heterozygosity, but homozygous null for BRCA1. As you can see at the bottom of the slide, those tumors arise too in breast cancer. They're not as common as, they are more common, than the heterozygotes by a lot, but nonetheless, they are important, and they're typically triple negative and basal. A few of them are luminal, and why there is such a tilting toward the non-hormone containing and expressing a form of BRCA1 breast cancer, big mystery, not well understood. Now, here's the first surprising finding made by Hua Wang when he was a postdoctoral fellow here in the lab and referred to his paper in 2019. You can see a series, not all by any means, of proteins, all of which we know to be BRCA1 associated. They include the Notch family regulator NUM, transcription factor, and perhaps carries out other functions as well. A real transcription factor called HES, which operates downstream of NUM. FANCD2, a major player in the Fanconi pathway of interstrand crosslink repair and early tumorogenesis suppression. CTIP, a BRCA1 associated protein that operates in the regulation of HR, or homologous recombination driven double-strand break repair. And these proteins, interestingly, when laid out in input fashion, that's what I'm introducing into a cell, you will see that we can eliminate them in the input if we need to. We could eliminate NUM, for example, as an exemplary case here. But now let's see what happens to all of them when we get rid of BRCA1. Or when we get, no, not when we get rid of BRCA1, when we get rid of NUM or HES alone. So if you get rid of NUM on the right, you can see NUM does not co-immunoprecipitate with BRCA1. But it also doesn't when you get rid of HES. And NUM and HES talk to one another, even interact with one another. Look at CTIP. Get rid of NUM, CTIP no longer co-IPs with BRCA1. Get rid of HES, CTIP doesn't co-precipitate with BRCA1. And if you get rid of FANCD2, NUM doesn't. And if you get rid of NUM, for example, HES doesn't. To make a long story short, if you deplete any of these BRCA1 binding proteins, and there are several others that elicit the same effect, none of them bind very well, even though the antibodies are directed at they're monoclonal and they're directed at a single polypeptide, as if one BRCA1 associated protein needs to be bound for others to remain bound, as if these proteins are talking to one another in the milieu as BRCA1 associated polypeptides. So you can ask the question, are certain BRCA1 bound proteins like commanders of the fleet, thinking about old stories of naval warfare, or are they capable of ceding that function to the commander-in-chief, which is BRCA1 P220. We don't yet know that, but all of the early suggestive phenomenology suggests that the latter may be true, whether it's actually commander-in-chief or it looks like or feels like commander-in-chief. But if you do the experiment in another way, you can find multiple commanders-in-chief to be determined. And then you can ask whether BRCA1 being an important player in all of this, being that all the proteins, each of the proteins that I showed you before needs to be bound to BRCA1 P220 for all the rest to bind, you could ask whether BRCA1 orchestrates the functions of other B1 or BRCA1, my local shorthand for the BRCA1 protein. You can ask whether BRCA1 orchestrates the functions of other BRCA1 bound tumor suppressing proteins like it, directing them to operate when and where they must in order to execute key functions, including cooperating, for example, in the suppression of any of a small collection of inherited cancers. And now we know even somatically generated cancers, which arise when somatic BRCA1 copy number is reduced either by 1 or even by 2. Now, here's a question that's based on real data. So about 6 years ago, Hua Wang, when he started his postdoctoral fellowship in the lab, observed that when any of the P220 subunits, BARD, NUM, HES, you name it, don't bind stably to BRCA1, or BRCA1 is missing from a mammary epithelial cell, that mammary epithelial cell dedifferentiates. I'll show you evidence in a moment. That means if BRCA1 is gone, or some of its bound acolytes, let's be presumptuous and say bound acolytes are gone, normal mammary diploid, primary even mammary epithelial cells immortalized with telomerase, they can't remain normally differentiated. As I'm going to show you, they acquire a mesenchymal phenotype. Moreover, we now know that in that setting, based on work of Hua Wang, Zhe Li, my beloved colleague at the Brigham and Women's Hospital, and multiple other members of our lab here, mammary epithelial cell dedifferentiation turns out to be an early step on the road to BRCA or breast cancer development. In short, compromising the function of P220, in fact, of a number of its bound proteins individually or collectively, may represent an experimental way of starting a cell off on the road to breast cancer development. So here's a little example. So we know you can get rid of BRCA1. Look down at the bottom. I'll see if I can put a little cursor on it. So you see this picture. Those are what are called CD44 low cells. They're primary cells that are immortalized with telomerase. They're diploid. They're even P53 wild type. They look normal. They form colonies. All is well. Now deplete them of BRCA1 and they undergo a dramatic change in morphology. Or instead of depleting them of BRCA1, let's expose them to an interstrand cross-linking agent like cisplatin, or mitomycin C, or acetaldehyde, or there are several. And now what happens is the same thing that happens when you get rid of BRCA1. The cells dedifferentiate to a mesenchymal state. Here's a semiquant on the top here in this figure up at the top, 3A. You can see that there's a dose-dependent change in the dedifferentiation process as you rise in interstrand cross-linking by cisplatin. And furthermore, you can see, look on the right up here, where you do a co-immunoprecipitation with antibody to various proteins, like, for example, N-cadherin. N-cadherin co-IPs with vimentin and actin. Notice that if you do the experiment, however, with two different mutant clones of BRCA1, you can barely co-IP E-cadherin, and there's a real problem with cell shape, as you can see on the left. as if there's something about the association of BRCA1 with certain partners, like E-cadherin, for example, other proteins that make up epithelial borders, which is associated with a normal cell state and which changes radically when BRCA1 has acquired the appropriate mutation, like in clone 4 or clone 51, which are clones that are BRCA1 all right. They encode BRCA1 all right, but there are mutations undoubtedly which promote the lack of E-cadherin binding and switch the protein over to one that binds vimentin, which is a hallmark of proteins that are involved in mesenchymal dedifferentiative changes. So, how does this work? I'll be quick at this. So we look down here on the bottom where we've imaged a series of linearly arrayed luminal progenitor mammary epithelial cells accompanied by the binding of a complex machine that contains BRCA1 and a bunch of bound associated proteins. I won't go into all of them just to save time. But this machine is outfitted either directly or through interaction with another protein or proteins, so that if you expose a cell to a sufficient dose of an interstrand cross-linking agent, even one as simple as acetaldehyde or formaldehyde, the cell responds by engaging in interstrand cross-link repair. The only problem is if it turns out that you deliver enough interstrand cross-linker, the repair is not enough, and the cell dedifferentiates, as you can see here in this tripartite drawing in which the central cell has changed shape and actually changes state of differentiation. Now that happens when there's trouble in the big BRCA1 central machine that's associated with chromatin. Now, if there's machine failure, this is showing you just a few subunits. There may be as many as 20 or more subunits in this machine. If you kill one or more that are essential for its function, you get the same effect, even without putting a mutation into BRCA1. For example, you see dedifferentiation, and now if you wait long enough, up to a year, one will see fully developed BRCA1 breast cancers. And if these luminal progenitors here were BRCA1 plus minus, virtually all of these cells would be BRCA1 null, having lost all of their capacity for using BRCA1 as a tumor suppressor. Now, this is a traditional finding in the literature. The clinical manifestations thereof have been widely heralded and accounted for in great detail. And there's only one other interesting fact that we want to bring to bear on this phenomenon, which is loss of BRCA1 function translates into dedifferentiation followed by tumorigenesis. And that is, if you look at the bottom of this complex slide at these micrographs that contain chromatin strands, and they've been stained with gamma H2AX antibodies. So we're looking for damage in a chromatin. And what you can see is that at 1 month, after you get rid of BRCA1 and allow the cell only to produce effectively no P53, no BRCA1, but everything else, you see cells now that are clearly have intact chromatin, but the intact chromatin at a month compared to the beginning of the process at the very time zero here on this graph, you start to see gamma H2AX staining at various defined places along the chromatid. Here it is at 1 month. Here it is at 1 month. You see a few spots. Look at 5 months. Now, like every third or so segment of the chromatid is red, staining with P53 BP1 antibody, a signature of its having been damaged. And by 10 months, you can see a lot of chromatid segments that are red. Now, if we look at nuclei, you can see that by the time you reach 10 to 12 months and not before, this is when there is now a gross tumor that is developed in the mammary gland of the experimental animal being analyzed in this case, every nucleus, basically every nucleus is red, means every nucleus is producing abundant p53, BP1 immunofluorescence, reflecting the existence of abundant ongoing DNA damage. It's interesting, we've looked at tens of these sections, and so far, when you get to a gross tumor and then wait another month or so, as the tumor enlarges, it almost seems, and I'm using the word seems, but that's the picture we get, that every nucleus is red hot with ongoing signatures of damage. And they're still there. And this is before the cells begin to die for any reason. They're still evolving as a tumor, as if there might be some magic in a pre-tumor cell acquiring enough damage that it is like maxed out on its ability to signal the presence of damage. And that may have something to do with tumorigenesis, is a reasonable hypothesis, which we and others, I'm sure, will be working on. And I'll come back to the kinetics in a bit. Now I want to speak about another body of work that begins to explore the relationships between BRCA1 and RNA molecules, which we'll show you in a few moments, and which came as a great surprise to us. And I want to highlight the work of Elodie Hatchie, a senior member here at Dana Farber, who is working hard on the problem of what the biological and biochemical significance of BRCA1 interacting with, much less talking to, much less being talked to by RNA actually is. So here's one of her experimental results that won't go away, as we'd say. So these bands here are all proteins that have been effectively immunoprecipitated with antibody to BRCA1 or not. On the left, we're just applying an extract to the gel. On the right, we're applying a BRCA1 IP derived from that extract. And please notice BRCA1 that's co-IPs with a variety of proteins we know it interacts with, and there are at least 5 more that we know of. Now, look what happens when you treat the extract with a ribonuclease, either RNase T1 or ribonuclease A, both of which are preferentially single-stranded RNases. And now what you do is you IP BRCA1 in all the lanes on the right. Please notice that where the ribonuclease was added, the proteins are gone. Like those proteins bound to BRCA1, maybe only when they were also bound to RNA. So the possibilities are that BRCA1 bound RNA makes BRCA1 like a magnetic multiple protein tethering machine, or that one or more RNAs binds to BRCA1 and each bound RNA tethers certain proteins to it, or there are numerous RNA associated proteins, each one or some of them, subsets thereof, bind to BRCA1 and of course to RNA, and or to one or more of the other BRCA1 subunits. There can be in these complexes that we co-IP as many as 20 or 25 or perhaps even many more BRCA1 bound proteins. Some of them may be RNA binding and use their RNA binding function to bring new to us, new polypeptides to the BRCA1 party, so to speak. And then you can imagine that there are soluble proteins that are freed by RNA cleavage. They remain immunoreactive. Now we know this to be a fact, with a monospecific antibody used to carry out the IP, and hence they were not fully denatured by RNase exposure. They're still immunoreactive, normally immunoreactive with multiple, actually multiple monospecific BRCA1 antibodies, 3. That strongly suggests that the proteins that in the normal state, in the unmanipulated state, bind to BRCA1, they don't not bind to BRCA1 because they become wholesale denatured. So that raises the interesting possibility that BRCA1 binding to a host of target proteins is an RNA-dependent process, possibly even a covalent RNA-dependent process, and we'll refer to that in a bit. So, importantly, BRCA1 is a DNA long known to be a DNA interacting protein. Ralph Scully was the first actually to demonstrate this both microscopically and biochemically. And it's also, interestingly, as we can see from this, a RNA binding protein, direct or indirect, that allows certain RNA species possibly to execute multiple new DNA repair functions, is a hypothesis. So let's chase this thought for a few moments. So Elodie Hatchie's work, recently published in this year, shows the following, all surprising. In a number of mammalian genes, there spontaneously developed single-stranded DNA breaks in transcription termination sites, and possibly even in transcriptional termination pause sites, transcription pause sites, which are near one another in these heterochromatic regions. And that's a form of damage. And the utter source of the damage is not entirely clear, but we do know that in the absence of BRCA1, it appears the damage grows. Well, Elodie and her colleagues have shown that these breaks attract BRCA1 and its partner BARD, most likely. We're not absolutely sure, but it seems that way. And then something quite surprising happens. BRCA1 saltates the formation of a complex involving it, undoubtedly a series of its bound proteins, and the three major participants in the RNAi machinery process, dicer and ago. BRCA1 dicer ago complexes form, and the end product of their action when fed with the appropriate messenger RNA, so far as we can tell in cells, is the processing of these RNAs to what she calls single-stranded RNA, single-stranded DNA-associated RNA that's 30 to 45 nucleotides, never been seen before. And this character, as she subsequently showed, forms a complex, actually saltates the formation of a complex that contains the RNA, which she calls sdRNA, PALB2, a professional known BRCA1 associated breast cancer suppressor, and RAD52, which is likely a cousin, at least functionally, of RAD51, the recombinase that's very important, homologous recombination. They form a tripartite, at least, complex under the influence, in fact, under the command of the appropriate sdRNA. And this machine, driven originally by BRCA1, now containing the RNA, which holds this amazing sdRNA PALB2 RAD52 complex together, promotes, if not engages in and directs single-stranded break repair at the two pause sites, in cycling, and even in G0 quiescent mammary epithelial cells. One has to wonder if this repair is in response to a defect, for example, in BRCA1 or some other tumor suppressor function. And if that were the case, is the outcome of this repair a tumor suppressing act? To be determined. Elodie and her lab are working very hard on this problem. We don't have an answer yet, but it would not be surprising to find that. Now, another colleague in the lab who's recently left to take up her science in Europe is Katerina Vahodina, who came to us from her home country of Estonia, and also by virtue of having spent a little bit of time, quite a bit of time in Belfast in Northern Ireland, where she did her cancer research training. And her work has led to a third surprise. So on the left, panel A is what happens when you carry out an immunoprecipitation with BRCA1, and then Katerina was fascinated by the behavior of telomeres. And she wanted to know whether telomeres talk to BRCA1. There was a paper in the literature from Professor Rosen's group at Georgetown. And so she decided to ask the question, does BRCA1 interact with proteins that live at, dine and support the formation of telomeres. They're called sheltrins, first discovered by Tizia de Lange. And what she found was that BRCA1 co-IPs virtually all of the known sheltrin proteins, TRF1, TRF2, POT1, TPP1, RAP1, and TIN2. There's still one more, but that's a pretty good majority. And when she studied the kinetics of the formation of BRCA1 binding to telomeres, she found that there was growing association through the cell cycle and a big jump toward the end as cells are getting ready to go back into G1 in M phase, as if BRCA1 has some, locum tenens-like right to interact with telomeres, as if maybe it's an important telomeric function. So she did a simple experiment and asked, was there some biochemical uniqueness to all this? And she and our colleagues in the proteomics unit here at Dana Farber carried out immunoprecipitations of extracts that we knew contained BRCA1, we knew contained sheltrin proteins because they were extracted from normal cells, and then asked whether the protein repertoires measured by MSMS that represent the TRF2, which is a major sheltrin protein. I'm going to talk to you a little bit more about this in a moment. Whether TRF2 interacts with a body of proteins, it clearly does. They're represented in blue. And BRCA1 interacts with a body of proteins, a large number of them. By this analysis, remember there were 250 BRCA1 associated proteins that have been formally accounted for, and 90 of them appear to represent equal binding partners of TRF2 and BRCA1. Now, what might that mean? BRCA1 is talking, spending time with, maybe even co-functioning with a major player, TRF2, one of a number of polypeptides represented in A above that plays a role in maintaining the integrity of telomeres. So what she did was, together with our proteomics people, carry out a Go analysis of the proteins that bound, all the proteins that bound to BRCA1, all the ones that bound to BRCA2, and she identified 15 of them that were most abundant, because you can see there are relative abundances in the middle of this figure here. Sorry. In the middle of this figure here, let me see if I can get my cursor on it. These are all the relative abundances. And 9 of the 15 different generic families of proteins, from messenger RNA 5' untranslated region binding, to messenger RNA binding, to ribonucleotide protein complex binding, and on. 9 of the 15 categories of proteins involve RNA. And BRCA1 and TRF2 reveal a propensity toward binding to, these are all telomeric proteins, dedicated telomeric proteins. 9 of 15 identified here, 9 of 15 groups identified here, not individual proteins. Groups are engaged in RNA binding. We were starting to get the message. Not only does BRCA1 play a role through the RNAi system in promoting the synthesis of an RNA molecule that executes key functional steps in DNA repair of single-stranded breaks, it's also got a lot of friends, maybe even family members, that are involved in RNA binding. And in fact, if you are willing to put up with this, unfortunately, too small slide, I still have a problem trying to identify very small objects and blow them up in a way that you can see them. But if you look here on the far left, so this is an experiment in which at various points in the cell cycle, Katerina isolated cells, made spreads, mitotic spreads from them, stained the chromosomes with antibody to BRCA1, used, for example, a probe for telomeric DNA, actually a sheltrin probe, which only interacts with the appropriate telomeric DNA, and also asked what happens when you merge some of the images, like far left here, where I can assure you that every arrow refers to a red BRCA1 and green telomere DNA structure interaction. So there are 4 clear sites. You can see some red over yellow staining in this one, maybe a little bit in that one, and in a number of other ones going down the line as the cell cycle evolves. And you can see that by the time at mitosis, when there's lots of BRCA1 and other RNAs bound to individual structures in the nucleus, including the telomeres, that there are a number of BRCA1 telomeric DNA structures that you can see. They form these red spots with yellow dots in the middle. So let's talk about telomeres for a few moments. We're talking about structures, of course, that have two ends. Those are the actual telomeric units. BRCA1 and polymerase II promote the synthesis of a product, a complementary RNA product from the telomere called tera-RNA. And furthermore, this tera-RNA, not only being telomere encoded, is synthesized in physiologic concentration under BRCA1 control. And you can see the same thing here as I just shown you, only the dots light up beautifully. But in this case, BRCA1 ploidy, wild type, heterozygous, and minus, that's its genotype, correlate with, as the BRCA1 abundance goes down, an increased number of spots that represent telomeres that are now lighting up because of a monoclonal antibody directed at order gamma H2AX. So this machine, which coats its telomeres with sheltrins, not only does so for the purposes of protecting their superstructure and even infrastructure, it also leads to ends that give rise to the synthesis of an abundance of tera-RNA. That's an RNA encoded by telomeric repeat units. And this RNA is a physiological product. And I've just shown you the sheltrin proteins bound to BRCA1 and shown you a hint that BRCA1 has something to do with suppressing damage at telomeres. Here's a better experiment. So what Katerina did here was take from primary cells, nuclei, and made spreads from individual cells from them, mitotic spreads, and then stain them for gamma H2AX in red, and in green for tera, which means staining for telomeric ends. So what she found was, if you have a hairpin directed at BRCA1 in those cells, but it's not turned on, you see very few red, yellow spot combined structures. But if you now get rid of BRCA1, the number of those structures is easy to see. It goes up. And that's the result of the formation in this setting of complexes, chromatin-associated complexes that have been shown by us and others to be loaded with RNA polymerase II, with tera-RNA, which forms a RNA DNA hybrid at this repeat telomeric coding unit. And that structure is RNA sensitive, being an RNA DNA hybrid. So if you add RNAs, this so-called R loop, which is an RNA DNA hybrid that gives rise in DNA terms to this DNA chromosomal unit here. I'm trying to get my, there you go. You see TTA, GGG, that's the DNA, that's the center of the DNA segment that's displaced by tera-RNA binding to its coding sequence. And it turns out that as you deplete BRCA1, the number of structures like these, which we call R loops, telomeric R loops, increases because BRCA1 depletion leads to an increase in tera-RNA, which we'll come back to in a moment, which leads to the formation of more of these telomeric R loop structures. And we could go on and I can show you repeatedly that if you add RNase H to these cells by transfection, the number of red, yellow dot structures goes down from a control where there's mock treatment. Add RNase H, many of these arrowed structures that are both red and yellow, reflecting the association of BRCA1 and tera, BRCA1 and the terminal ends of telomere of chromosomes disappear. And some proof of that is in this quantitation. You can see that these are percentage of cells that we analyze in these spreads with 5 or more co-localized gamma H2AX foci packed onto telomeres. And in the presence of RNase H, as these structures increase in the presence of RNase H, their number is greatly reduced, reflecting the fact that their existence is dependent upon RNA-DNA hybrids. So I'm going to go quickly because I want to get to the ultimate end here is BRCA1 is also a suppressor of replication stress, of replication-associated damage. It is a director of repair. And so if you analyze these spots of the type I talked about before, hunting for terra spots and asking whether or not they are bound to reporters like phospho-ATR or p53BP1 of DNA, shall we say, replication stress associated damage, it turns out that they are, and you can see more of these red, yellow spotted guys every time you get rid of BRCA1, like BRCA1 is responsible for suppressing telomere-associated replication stress-driven damage. And you can even see that some telomeres kind of look abnormal. They've undergone the normalcy that is associated with telomeres, which should be perfect, almost perfect copies of one another, is that BRCA1 not only suppresses replication stress, it also suppresses telomeric disorder. And we can find that if we want to know what happens, for example, to the RNA production from a telomere, we could ask what happens, for example, to the binding to telomeres in the presence and absence of BRCA1 or certain phospho forms of Pol2. S5P, Pol2 is a guy that's involved in the initiation of transcription, S2P, a guy involved in the continuation of transcription. And interestingly, if you want these guys to go away, you kind of need to be sure there's BRCA1 there. As if BRCA1, as I told you, which is a suppressor of terra transcription, and this is a terra probe being scored for here. In the absence of BRCA1, there's abundant terra RNA, but in the presence of BRCA1, that's SI control, there's much less of it because Pol2 function has suppressed the transcription. So what am I telling you very quickly here? And then I'm going to move quickly into the last part of this talk. In the normal setting, when there's plenty of BRCA1 available on telomeres, as I showed you, you can find it on telomeres, there's a suppression of transcription of telomeric DNA by Pol2. As a result, the number of terra R loops that form, remember that BRCA1 represses terra transcription, limits the number of terra R loops, and a terra R loop induces DNA damage, local DNA damage. In that case, there's relatively little terra present, and the genome pretty much stays intact. If, on the other hand, terra transcription is allowed to blow up, if BRCA1 is gone, there is a great abundance, even in excess of terra RNA, which forms terra R loops, which are damage-driving structures, and that leads to a variety of abnormalities. Not only increased terra transcription, increased terra R loop formation, right? As I've shown you before. Let's see if I can get my cursor on it here. Here we go. Here's a terra R loop. There's an abundance of those guys. There are free chromosomal ends that are not protected by telomeric sheltering proteins, and there's increased telomeric DNA damage. So now we're going to get close to the end here. So along the way for carrying out these experiments and having seen a bunch of times that BRCA1 parks itself at telomeres, as I showed you about 10 minutes ago, Katerina asked a simple question. If it parks itself at telomeres and so does BRCA1, do they co-park? they park together? Do they interact with one another? To make a long story short, she used an old technique developed by Junji Chen in our lab many years ago, in which you can cleave BRCA1 proteolytically into 6 fragments. You can see them here. See if I can get them up here for some reason. I got to go back. All right, you can see the 6 fragments here, F1 through F6. To make a long story short, Katerina asked, if I take wild type, an oligonucleotide, a long oligonucleotide that represents wild type Terra RNA, and expose it to each one of these 6 fragments here. I can't seem to get, here they are. The 6 fragments, each one is coupled to GST. You incubate it in an extract that contains wild type DNA. Wild type DNA, for example, from someplace in the genome, I'll show you in a moment, for example, from this fragment F2 or this fragment F3 versus F2 with a mutation, the nuclear localization, a nuclear localization sequence. See this red thing here is present here, missing here, and a mutation in it, a single point mutation in it, which comes from the lung cancer of an individual that was discovered in Britain, were all incubated with, for example, GST-F2, because in the screen that she carried out, she asked whether each one of these fragments of BRCA1 interact with these RNAs. And the answer is that, with these DNAs. What she showed was that the F2 fragment can interact with this particular segment, the N terminus of the F3 fragment, because it's a duplicate, this sequences, can both interact or each interact separately with this nuclear localization sequence of about 20 amino acids. Now, interestingly, if you take the wild type sequence from the end of F2 and introduce the elimination of the nuclear localization sequence from this red stripe nuclear localization sequence here, or put a point mutation into it, notice that the ability to bind, this case, Terra RNA, goes down in the case of both mutants compared to the wild type. And that's reproducible. I'm going to get past this pretty quickly because I don't want to burn up all your time. But suffice it to say, at this point, Katerina found that either BRCA1 depletion or either of these nuclear localization sequence mutations that prevent Terra from binding to BRCA1, because that was the question here. Does Terra RNA bind to these nuclear localization sequences? What she found was they do, and that binding depends upon an ability to recognize this particular nuclear localization sequence, this red stripe right here. So you've got a sequence here in the genome capable of binding to this nuclear localization sequence. And what you can show is that ribonuclease suppresses the development, for example, of the damage that arises here when there's no BRCA1. If there's some BRCA1, you see fewer of these red-yellow spots, and in the presence of wild-type BRCA1, they're normal. And I won't go through the detail, but suffice it to say that ribonuclease H is preventing these spots from forming, which means that RNA-DNA hybrids underlie the formation of all these damage-reflecting red-yellow structures. And if you go on, you can show the same kind of phenomenon. If the wild-type BRCA1, for example, is wild-type versus it has a nuclear localization deletion in it that's sizable, 6 amino acids or 7 amino acids, 6 amino acids. And in this case, if you have a point mutation in that sequence, in the nuclear localization sequence of interest, you can see that when either of these 2 mutants confront these spread chromosomes, there's, as you see with these arrows, trouble in the formation of normal telomeres. And you can analyze this quantitatively with the same result every time, as if BRCA1 is there to do something at a chromosome, at the telomere of a chromosome. And in fact, the question was whether tera-RNA actually binds to those sites. And as you can see a couple of slides ago, that's what we observed right here. These are tera-RNA pull-downs by wild type or either of those two mutant sequences, each of which in the mutant case is defective in binding to the end of either F2 or the right-hand end of F3, which are DNA sequences. And so the question is, what might that mean? There's a problem with BRCA1 being able to bind to tera-RNA when there is a hole in the sequence we've been talking about, which is a sequence that's part of a nuclear localization sequence. And just to summarize, if that hole persists and binding is poor, and what happens instead in the absence of BRCA1 is that telomeres and these nuclear localization sequence units begin to fill up with damage. You can heal a lot of the damage by putting back wild type BRCA1 in place of the mutant or in place of the absence of it. But when you put back either of the two mutants, the guy that's missing 5 amino acids or the guy that's got 1 amino acid substitution, both of which are mutant, the damage containing telomeres reappear. So I'm going to come right down to this now. So tera binding to BRCA1 P220, which Katerina analyzed using fragments of BRCA1. That's how she knew that tera bound to a specific sequence at one end, at the C-terminal end of a fragment of a few hundred amino acids, a specific tera interaction to be determined to exist. Even a point mutant in that sequence was enough to kill tera binding. And when all is well, one can show that tera binding to that sequence correlates with the suppression of R-loop DNA damage at and near that site in the chromosome. It correlates with replication fork maintenance, that is a suppression of replication stress, an example of which I showed you a few moments ago, and is associated with normal telomere structural maintenance. The ends of chromosomes don't get eaten away. Now, these are phenomena that when suppressed, it is possible to hypothesize that suppression of each one of these abnormal outcomes, not the normal ones shown here, but the abnormal ones that occur in the absence of tera-RNA binding to a very specific site on BRCA1, might be defective in the suppression of certain neoplastic outcomes. After all, there's damage at each one of the binding sites when the local recognition site for tera is defective. So the argument then is, and I'm nearly finished, that when the tera binding domain on BRCA1 is mutated, and tera cannot bind to it normally, endogenous tera cannot bind to BRCA1 normally. Endogenous tera, whose synthesis is regulated by BRCA1, whose transcription is regulated by BRCA1, aberrant telomeric structures develop. Replication stress develops. Telomeric DNA damage develops. And the full outcome of that we're studying, but one wonders about whether, given the fact that these are telomeres, one outcome is senescence, which is, of course, as shown by Judy Campisi some time ago, and beautiful papers since that time gives rise to the so-called SASP, cytokine production phenomenon. Some of those cytokines may induce aberrant changes in the structures and formation of certain key organelles in cells. They may promote dedifferentiation, one of the early steps on the road to generating a real BRCA1 breast cancer, and they may even be involved in suppressing the development of tumorigenesis. So that's the big question. If a little RNA bound to a tiny site on the surface of BRCA1, which we know is important to suppress R-loops, R-loop damage, replication fork associated damage, which means replication stress, and telomere structural maintenance. All of those go south when TERA can't bind to that site. So we wonder, we don't have to wonder. There is a major role, I think, established in many labs for BRCA1 failed synthesis for triggering chronic DNA damage, followed by basal to mesenchymal dedifferentiation on the road to triple negative breast cancer development, where, as I'll show you here, last data slide, you'll see in the tumors of these patients, every single nucleus is red, having been stained with monoclonal antibody to p53BP1, a DNA damage reporter protein, and a participant in DNA damage control via non-homologous end joining. Every single nucleus that we could analyze, we analyzed a number of them. Over time, by the time there was a tumor, every single nucleus was red. It's not like some were red, all were red. And so one wonders whether, like almost global tumor cell damage of a certain type predisposes to events that allow the tumor to grow. We have no direct evidence for that whatsoever, but it's a question well worth asking. And I'm just showing you in this particular slide here, a simple slide here, work from Hua Wong, that by the time every cell is red, the tumor is long ago established. It's almost like the tumor response to DNA damage may be helping the tumor to maintain its normal abnormal architecture. Who knows? To be determined. So finally, can BRCA1 prevent aberrant cell development, at least in part by maintaining stable sdRNA, the small non-coding single-stranded RNAs, maintain their interactions with some or all of BRCA1 tumor suppressing, BRCA1 associated tumor suppressing proteins like BRCA2, PALB2, possibly RAD52? Or is it interacting with RNA so that some of its other DNA repair and epithelial differentiation promoting subunits continue to bind to it when they're needed functionally to maintain some major repair or post-repair event that keeps a cell alive after DNA damage has had its way, either before tumorigenesis, during it, or afterwards? And finally, for maintaining physiologic levels of TERA-RNA and TERA-BRCA1 binding, as I've just shown you, which in turn help maintain telomere integrity and inhibit telomere replication stress. Now, perhaps the most important thing I can tell you is that those of us involved have had the privilege of working together in a wonderful group of scientists, including Elodie Hatchie and Katerina, Serena Landini, Liana Goring, Derek DeConti, Fida Abarazak, wonderful young postdoc in the lab, Vlad Bochkarev, Amy Liu, Mai Hin, Andrew Lee, wonderful scientist at Dana Farber and in the lab, Liz Murphy, and Brenda Chen. I thank you for your attention, and I think we're finished pretty much on time. I'm sorry I had to cut out a few slides, but I didn't want to run over it. Thank you. Be glad to take questions.
Thanks, David. What a wonderful and stimulating presentation, and really remarkable work on RNA interactors for BRCA1 biology. We have several questions, and if we are not able to make it through the list by the end, I think we have a mechanism for emailing you the questions so that you can respond in your own time to the questioners. But our first question is from Steve Elledge, and regarding the ICL induced EMT phenomenon that you observe and its relation and its interaction with BRCA1. He's asking, is the dedifferentiation in response to ICLs regulated by the DNA damage response and by ATM or by ATR?
We only have preliminary data that address that, but let's simply say in association with these dramatic morphological changes, there is activation of ATM and ATR. Now, the extent to which they feed the dedifferentiation is sadly to us as yet unknown.
Okay,
Although I should say, since we can make the damage lead to dedifferentiation simply by applying to a cell a small molecule that induces a form of damage of the type we've talked about, there is at least an intimate relationship between the damage and the outcome. How direct the phenomena are in their connections, I don't know yet.
So if you treat with an ATR inhibitor, do you see any change in the sort of pattern of dedifferentiation?
Embarrassingly, not yet done.
Okay. All right. Our next question is from Natalie Vaninoff, and she's asking about R loops and relating. So the question is, what's the relative proportion of R loops that are intermediates in an HR function versus Terra R loops? And she's curious about how stable the Terra R loops are at telomeres versus those R loops that might be related to intermediates of homologous recombination.
So that's a good question. I can't answer it directly, but we can generate R loops in cells kind of on command by either getting rid of BRCA1 or something like that. And those structures hang around for days.
So quite long-lived.
Yes, seem to be. Now, there are ways of dissolving R loops. BRCA1 binds two interesting proteins. One, I'm blocked for the moment. It's an endoribonuclease. I'm forgetting the name for a moment. And its job is to eliminate the bound RNA of an RNA-DNA hybrid. Also, senotaxin promotes the dissolution of R loops. The extent to which quantitatively these effects take place is still a matter of experimentation.
Mm-hmm. Okay, and I think the final question, we'll send you the remaining questions because we are, I think, now at the end of the time, is from Dr. Stephane Richard. And the question is, do you know what the RNA species are that bridge BRCA1 to SETX, DICER, AGO1, BARD1, etc. in the figure you showed?
Under intense investigation by LOD and others. And,
right.
Do
you see this as being a sort of DS RNA function, or do you think a different RNA species type might be involved?
Well, we don't know enough about the structure. We don't even know quite how heterogeneous these RNAs are, although we have a hint from sedimentation velocity size analysis experiments that it's not just one RNA doing all this, at least not in length. But more than that, I can't say, Ralph, because we just haven't analyzed those RNAs carefully enough. But we will know. It's a major project in the lab.
Okay. So I think we are out of time, but David, thank you so much for a wonderful seminar, and we will forward the remaining questions to you afterwards so that you can be in touch with the questioners.
Thank you so much for everything, and I thank the organizers of this seminar for their tolerance. I apologize for the weak moments or two towards the end. Thank you.
Cool. This was really great, and reminder to you all that this is the last presentation in this year's seminar series. We'll be starting up again in the fall, so look out for emails regarding our next set of speakers, and have a great summer, everyone.
Thank you.
Okay, bye.
Bye-bye.